Hello everyone, I'm Subalakshmi, first MSc Microbiology, Sacred Heart College Autonomous. My topics about space microbiology. What we are going to see in this page: introduction of space microbiology, historical content, space environment and microbial adaptation, microbes in space, microbial growth in microgravity, current research in invented, application of microbiology in space, how does space affect microbes. Introduction to space microbiology. Astromicrobiology is also called as exomicrobiology, is the study of microorganisms in outer space. It stems from an interdisciplinary approach which incorporates both microbiology and astrobiology. Astrobiology effects are aimed at understanding the origin of the life and the search for life other than on Earth. Historical content. Yearly exposure to space. In the year 1970s, the Apollo 16 and 17 mission exposed microorganisms to space conditions for the first time. The microorganisms were exposed in the MEED, Microbial Ecology Equipment Device, facilitated and in the biostat experiments. Space environment and microbial adaptation. Microbial adaptation to specific space flight condition may lead to alternation associated with energy source utilization. Hereby a corresponding molecular pull up from energy converting enzyme responsibility for a switch from one energy source to another is affected in condition of space flight and stimulates microgravity. Three basic theories for the research of space microbiology. Hereby there is have an space environment up to this space environment. Here microgravity, low nutrition, space radiation, high pressure, extreme temperature. Microorganism mutation are divided into three steps, includes pathogenicity mutation, virulence mutation, mutualism with humans, astronous production, drug resistant mutation, metabolic production mutation, space corruptions, microbial contamination, space microbial pharmaceutical, spacecraft production and new materials. Next slide. Microbes in space. Microorganisms that have been founded in space including bacteria, fungi and archaea. Some of the bacteria includes Staphylococcus and Malaise, the most common bacteria and fungi found in the International Space Station ISS. Then Enterobacter buchantensis, a multi-drug resistant bacteria that coexist with other microorganisms on the ISS. Then Aspergillus species and Penicillium species, fungal species that have been founded on the ISS. Saccharomyces cerevisiae, a fungus that exhibit genomic rearrangement to survive in the stressful condition. Bacillus subtilis. It is a bacterial spore that have been trusted for survival in outer space. Microbial growth in microgravity. Microbes can behave differently in microgravity conditions, such as in space, that they do on Earth. Growth. Some bacteria may grow more in space and may even be able to grow in the presence of antibiotics that would normally inhibit them. Virulence. Some bacteria may become more virulent or able to cause disease in space. Drug resistance. Some bacteria may become more resistant to antibiotic in space. Biofilm formation. This biofilm formation, microbes may form biofilm differently in space. Ge- expressions. Microbes may experience change in gene expression in space. DNA damage. Evaluated radiation level in space can damage the DNA, which may activate SOS response system. Microbial contamination in space can be a serious health concern for astronauts and can impact their safety and performance. Current research initiative. NASA and other research organizations are currently conduct a number of space microbiology research. Invites includes microbial tracking. NASA's microbial tracking experiment series studies microorganisms on the International Space Station ISS. The series includes collecting samples from the station's surface, air, and water, and studying bacteria, fungi, and virus. Closed habitats. NASA developed closed habitats with microbial cultures to study how bacteria grow and evolve in space. Microbes in human health. Microbes in a renewable resource. First, microbes in human health. Negative impact. The harsh environment of space can cause microbial biofilm formation that will corrode equipment and promotes antibiotic resistance and virulence. HGT can increase pathogenicity and biofilm formation. Mitigation of negative impact. Specialized surface like copper, silver, and titanium and sterilizing treatment using chemicals, UV light, or sound waves can help detect microbe growth, HGT, and biofilm formation. In situ resource utilization. Microbial fuel cell can generate energy from solid water, plant decay, and solar radiation. Microbial biomining can excrete metal from lunar or marine surface. Biogenerative life support. Microbes provide extra nutrient for plant growth and help break down solid and liquid waste from resource and water purification. Photosynthetic microbes efficiently convert CO2 to H2O and O2. Engineered microbes. Engineered microbes can more efficiently produce antibiotic nutrients and biomaterials and be used for drug delivery, disease detection, and radiation schedules. Applications of microbiology in space. The microbial species present on a board that ISS or its microbiome may provide information that is helpful to the prevention of disease among crew members by identifying potential harmful organisms, as well as areas of the spacecraft that are potential hotspots for this pathogenesis. How does space affect microbes? Most bacteria are harmless and may are beneficial or even essential to human functioning and well-being. But microgravity can make some microbes more likely to cause disease, and bacteria and fungi may affect the function of spacecraft system by, for example, corroding metals. Apart from this PPT, American Society for Microbiology ASM is committed to maintaining your confidence and trust with Respect to the information we collect from your own website on with operated by ASM and other source this privacy policy set for the information. Apart from this subject, plenty of microorganisms in the world and out of our world. Thank you one and all for your patient listening.